Thank you very much for giving me the possibility to speak here at this meeting. And you see, I will talk about the use of sequenced coevolution for protein structure prediction. And there were quite some talks about these topics already in this meeting. And I changed a bit my presentation in reply to some of the questions which were asked in these talks. But I hope this will work out nice. Okay, what is the main idea? The basic idea is that we have a lot of sequence data available now and much less structural information, but in evolution, typically structure is much better conserved than sequence. So you have a lot of sequence variability with having relatively conserved structure. And so somehow what we would like to do with using the huge amount of sequence data is to use statistical inference to use the sequence variability to inform structure prediction. There are two very little ideas which are somehow going into this inference. The first one is, the first observation is that residues which are in contact in a folded protein, like here in this cartoon, these two, typically undergo coevolution. So this means they undergo coordinated changes in evolution, and which leads to some measures, some kind of correlation in multiple sequence alignment. So in the variability of the sequences, we have some signal about contacts. So what we would like to do is to go back to do statistical analysis to discover contacts. So that's not at all a new idea, and somebody who has been working about this just came into the room, one of the authors of this paper. And anyway, there is a second, and the correlations are very easily measured. For instance, one way is just that you look to two columns, and if you want to observe correlations, you just look to the statistics of amino acids in each of these single columns of this alignment, and then you look to the joint occurrence of amino acid pairs in the same protein in the two columns, and then you see if this Fij is different from Fi times Fj, it's correlated. It's very simple. But there's a second input which is crucial to get good information out of this correlation, which is correlation is not coupling. So you can have correlation resulting from coupling, yes. But you can also have correlation which is mediated indirectly via some intermediate residues. So you don't have any strong coupling between these residues I and G here, but you have some intermediate coupled residues. And somehow, intuitively, this one case here might correspond to a contact, the other not. So can we somehow disentangle the two cases, direct and indirect? Can we somehow, and obviously we cannot do it by just looking to I and J at a time, but we have to look to them in the context of all the other residues. So we have to do something, or one possibility is to set up some kind of full-length sequence statistical model. So this A1 to AL is a full length of a sequence. So it's one of the rows of my sequence alignment, basically. Okay, the minimal model which actually captures covariation would be a Markov random field, or in statistical physics, which is my background, it would be a Potts model. And basically it looks like an exponential of a sum over pairwise terms and local terms. And these pairwise terms are the direct couplings we are after. So these would be the red lines in the slide, 2 slides back. We want this, obviously, we want to give numbers to these direct couplings, so we have to fit these parameters such that the model is coherent with the data. Coherence with data in particular means that if I calculate marginal distributions for just 2 positions, I should find what I counted in my data. So I should just recover these empirical frequency counts which I introduced before. Obviously, this sum is exponential. So if you want to do this exactly, you would need something like 21 to the length of the protein. So it's completely impossible. So you have to do something approximate, and there is now a whole market of approximations out there. some in collaboration with myself, some by other groups, and they have all advantages, disadvantages, and so on. And I don't want to go into the details of this technical, the technical details. I will at a moment talk about this, which is a paper which is not yet out and which answers some question which has been talked during this meeting. Okay, what is common to these methods is basically that they strongly improve contact prediction compared to correlation. So here I give just one example, the trypsin inhibitor. I introduced all pairs, the 30 strongest correlated pairs on the left and strongest coupled pairs on the right, and I colored them green if they are not in contact and red if they are in contact, and you see direct couplings are better. Okay, this works actually across many protein families. Hundreds of protein families have been tested. It works not always as well as in the trypsin inhibitor, but you have always an improvement. But it's abundant protein families, so we need at least about 1,000 sequences. And there was always, in all talks about coevolution here, there was the question, why 1,000 sequences? Why do we need so many sequences? And actually, the question is ill-posed in a sense, because if you just do a little counting, how many parameters we have? I have introduced these coupling matrices, E, I, G, A, B. So I, G are 2 positions in the protein, A, B are 2 amino acids. So these are 20 L was the length of the protein, squared parameters. So if you just count for a protein of 50 to 500 amino acids, you have something like 1 million to 100 million real valued parameters. Now look to an alignment of the same protein length with just 1,000 sequences. You have L times M times log 2 bit of information maximum, which is 200,000 to 2 million. I fit 100 million parameters with 2 million bit. So why does it work at all? So the question is not why do you need so many sequences? Why can we do with so little sequences in a sense? But anyway, the idea should be somehow to reduce this number of parameters. And basically, there are 2 main methods to do this. One is, would be to try to sparsify this matrix, to put as many elements as to 0 as possible. That's actually what David Jones' group in PsyCov does. But for this DCA, direct coupling analysis, it turned out not to work well. It immediately degrades the accuracy of prediction. So we came up with something a bit different, which is dimensional reduction. So we describe this coupling matrix by a finite rank matrix. So it's a bit in the spirit of principal component analysis, where you replace a covariance matrix just by a few principal components. So these we have these we write it as in this quadratic form via some position-specific patterns, and these patterns, they look a bit like position-specific weight matrices, and actually they describe directions and patterns in sequence space, and they can describe either the directions which are actually enriched in sequences or which are deprived of sequences. And actually the deprived one for our are the ones which are more important for structure prediction. But I can't go into this. We select patterns by maximum likelihood. So you can somehow, it's very technical, and the paper will come out in a few weeks, so it's in production at the moment. So concerning to this number of to this question of the many sequences which we have or do not have, we just did a little experiment here and looked first to the 2 full lines, the black line and the red line. It's it's an average over several protein families. We plot the true positive rate for contact prediction as a function of the number of predictions. And obviously, we would like to have a curve which is up here. And you see for a large alignment, 1,000 sequences, we have a curve which starts nicely. For small alignment, 50 sequences, we have a curve which is down there, which is basically random, so which is flat. we have no signal in there. And that's the full DCA, so the one with the 1 million to 100 million parameters. Now, if you reduce the number of patterns to, say, 200 or so, basically nothing happens. It's the same result. Now, if we reduce the number of patterns, so the rank of this matrix in the last slide more, so for instance, to 128, you see that for the large alignment, the performance starts to degrade. But for the small alignment, we start to find some signal. We even more reduce this dimensionality, 32 patterns, and you see it degrades more, and it comes better down here. And then for too small alignment here, it's very much degraded, but here again starts to degrade. So there seems to be some optimal thing. Obviously, true positive rates of 50% at the beginning is not a great deal, but we are talking about an alignment of 50 sequences here. And so the common standard, what we say, is that we need 1,000 to get the signal. So this is not an answer to the question how we can really do with less sequences, because it may point into a direction where one can go to get closer to this, to answer this question. Okay, back to standard direct coupling analysis. And so what you observe is not only that the prediction of contacts, the number of contacts predicted is better from direct couplings than from correlations, but also the kind of how they are distributed. If you look to contact maps, so the gray one here is a native contact map, so this is what you would like to produce. And the red points are predictions which are on top of a gray point, so true positives. The green ones, again, false positives. And you see that in particular, long-range contacts are not captured just by correlation, but by couplings. It's sparsified, but still it can guide structure prediction. And you have heard very nice talks by Debbie Marks and by Tim Nugent 2 days ago, or some of you hopefully have heard these talks. And they have done a very great work using methods like this. So in one case, this method, in one case, the related Psychoff method, to predict even large transmembrane structures. So my contribution to this is much more modest, but I would like to discuss this one, the specific case, because I like it. It's a nice work I like pretty much. And it's a work which took us quite a long time, because we didn't want only to predict, but we wanted to use our predictions also to experimentally verify. So, and this took us to a scale of 4 years before we got something published, but last year finally it came out. So we concentrated on one specific system, a histidine, sensor histidine kinase from bacterial two-component signaling. So it's a transmembrane protein, and if signal is received by the sensor domain, which is extracellular, then this histidine kinase domain auto-phosphorylates conserved histidine residue, and then this phosphoryl group gets transferred to a response regulator, which gets activated typically as a transcription factor. So it's a very simple signaling system, and by bacteria it's used over and over, this architecture, for very different signals. That's why already in 2008 we were able to collect 13,000 sequences for these two domains, histidine kinase, HATPase together. And I want to talk about the interaction between these two domains, histidine kinase, HATPase. 13,000 sequences, many of these systems essential for the bacteria they're in. In 2008, one single structure, this one, in an inactive configuration. Inactive means that the ATP is very distant from the histidine, which should be phosphorylated. And while we were working on it, two more configurations appeared, two more structures appeared, and all of them being in the inactive configuration. But we have these 13,000 sequences. So just let's do the analysis, as I described you before. And with this analysis, we found 5 interdomain contact predictions. 5 because we restricted to the range where the intra-domain prediction was highly precise. Okay, we have these 5, and 2 of them are actually in contact in this inactive structure, which I showed you before, 3.9, 3.3 angstrom, and 3 are distant. So we get 3 predictions at maximum for this active configuration, and actually when we look to them, when we map them onto the structure, it looked pretty nice because we have these 3 here, these 3 residues here, and if you bring them close together, you seem to bring also the ATP close to the histidine. So it looks nice. We have just 3, it's very little, but anyway, they look nice. Okay, this motivated us to somehow try to guide molecular dynamics simulations to actually get a prediction for the active structure. So we took our predicted contacts, we took the inactive protein structure, and in the first step we used some very coarse-grained Go models to guide some kind of docking simulations, which gave us, as you see here, you see some results overlaid, basically an ensemble of structures, rough structures, which might be close to the active structure. And then we took this ensemble and we tried to refine in a realistic force field without using these results. So just the structure, yeah. And without using this. So, and some of a part of this ensemble actually stayed stable and gave us this structural prediction. Okay, I told you that we want, no, just one more remark. You see here the inactive and the active structure overlaid, and it's actually a very large conformational change. So basically this H-ATPase domain is rotated by almost 90 degrees around the histidine kinase domain, and probably without these, the help of these 3 contacts, we wouldn't have found this structure. I told you we wanted to verify our results experimentally. So let's look to the contact map, which comes out of the final MD simulation. So here the interesting part are these off-diagonal rectangles, which are the contacts between the histidine kinase and the H-ATPase domain. And you find in this, you find well the 3 contacts which we predicted in clouds of contacts, so this is nice. But there are some clusters of contacts here between this helix and the histidine kinase domain, which are not captured by the method, by the statistical methods. So we decided to concentrate our experiments on these 2. And what did we do? We took a nice system which is experimentally accessible, gives a nice phenotype, sporulation kinase from B subtilis. And in the first place, we produced a non-functional hybrid, which has 1 domain from the histidine kinase domain from a kin-A and the H-ATPase domain from kin-D. And it's not functional. I will show you the results in a second. And then we used our structural prediction, because we have predicted an interface, to exchange the residues which are in the predicted interface to fake either the interface of the kin-A or the kin-D. So 2 possible experiments. And if our prediction would be correct, maybe we can reestablish sporulation activity. See what happens. So these are in vivo data, some in vitro data. In vivo, you see if the kin-A is present, cells are growing. It's a white bar, and they are sporulating. It's a gray bar. If you delete the kinase, they're growing, but there's no sporulation. The same with the hybrid. So it's growing well, but it's not sporulating. Now we have the 2 possible reparations, and the one repair is just re-establishing basically wild-type sporulation activity, and the other one is re-establishing something like 1% only, but before we had this. So it's anyway 6 orders of magnitude above, and we have just repaired a part of the surface. I told you that we just concentrated to the contacts on this alpha-3 helix. So still this is pretty nice. And just as a last thing, we even tested the single mutation, and some came up with a very strong effect. And we were asking ourselves, so if we have this strong effect, why didn't we see it in a statistical analysis? So in one sense, this is very nice because it seems to confirm our results, but on the other hand, it seems to invalidate our method because there is strong effects and we don't see them. So we looked back to the data and was quite nice to discover that we have used the PFAM definition of the domains, and the histidine kinase actually covers all the histidine kinase, but the H-ATPase domain, as defined in PFAM, which exists in very different proteins, does not contain this alpha-3 helix. So it was not in our data. So how could we find something which was not in the data? And so we realigned a full-length alignment, and we found two more contacts, which are this one here. And at this moment, we were kind of very happy and convinced that this was actually the right structure. And actually, half a year after we published, a new crystal structure, the fourth one, appeared, and it's a funny one because it's an asymmetric structure, which contains the inactive here and the active conformation. And actually, in the inactive, the two inactive predictions are in contact, and in the active structure, the active predictions are in contact. So which beautifully confirms our prediction. Okay, I want to thank many people, and in particular, in blue, I noted the tight collaborators, and I should mention in particular Henrik Surmand, who is a biochemist, who is responsible for all the experiments, and Alex Schuck, who is able to run all these molecular dynamics simulations. Thank you.
Thank you very much. We have time for a few questions. Feel free to just, we don't have a
So in statistical learning, there is a group of techniques like structured output prediction. There is a group of ANTA-SCAR. They're basically methods that can directly predict the structure, for example, structure of interactions or structure of proteins and stuff like that. Do you think this could be applicable to this problem, or are you aware of these techniques?
I'm not aware of these techniques. Okay. So the closest methods in the statistical learning community to this are actually graphical model learning techniques, which basically have been applied. So some of the methods, which the pseudo-likelihood methods, directly come from the statistical learning community, but these are just learning graphical models, so without this three-dimensional structure or so below, but the other thing.
I think there are probably techniques that could be applied for such a problem.
There are probably many techniques, yeah.
There's another question.
Very nice talk, thanks. Can you give an intuition why these lower rank matrices work better in constraining your problem than the L squared prior or the L1 prior that, for example, in cyclofuses?
Compared, I don't know why the L1 in this case does degrade immediately the accuracy. We tried in several ways, and it didn't work out very well. And I don't know, maybe we should discuss this because you use it and it works. And several people tried, and the question why this low rank should work is, again, parameter reduction. And you can, you can relate these patterns I was talking about to the spectral properties of the Pearson correlation matrix. So basically, these patterns are some transformation of eigenvectors of the Pearson correlation matrix.